Okay. Still thirsty. We are in the middle of talking about event-related potentials, and in the last session we talked about one class of event-related potentials, which we call the sensory event-related potentials. You can see them a little bit here. So we talked about the visual evoked potential at some length, also the somatosensory evoked potential at some length, and then the auditory evoked potential a little bit short because time was running out. But essentially we went over them, and your task is now to know what kind of standard paradigms are used for that, when the effects occur, and essentially where they occur. But I think this is relatively straightforward from your basic knowledge of functional anatomy, and the rest is just memorizing. So today I want to talk about another class of event-related potentials, which I call here cognitive event-related potentials. I might also want to call them surprise event-related potentials, but we will only see that later on. And the ones that I want to cover are mismatch negativities, the P300, language-related event-related potentials, error-related negativities, and then finally we'll also look into event-related potentials that relate to movement. So all of these are used heavily in cognitive neuroscience. So I personally worked on mismatch negativities and P300s because there's currently like a very small subfield or a very small collection of people who actually look into the computational modeling of these, and like every year or so, or let's say every year you have 2 or so papers that do that. I'm also working in a collaboration with Milena and one of your colleagues, Leah Muzirulek, now working on some language-related event-related potentials. So a little bit of first-hand experience with these. I don't have first experience with error-related negativities, also not with movement event-related potentials, but they exist, and they are people involved with SCAN work on them, and the whole world of cognitive neuroscience works on them. And they are interesting. So, has any one of you done EEG? And was that related to any of those?
Any of the ERP?
Yeah? Yeah, okay. And you? Also?
ERN and N2PC.
Sorry?
ERN as well and N2PC.
N2PC.
Like attention related to
This is actually, there we see there are more specific potentials that people look at. This is just a selection. So there are, maybe at one point I find time to actually put some more in there, because of course the ones that I teach, these are the ones that I know well. The other ones I might have heard of or not, and have not really investigated myself. Okay, so these things exist. Your colleagues provide a reference, and I'm also telling you that they exist. Or at least people study them. So for each of these, and this was the slide that I was looking for already when we talked about event-related potentials, it helps to structure the whole thing and not get lost in all these abbreviations. And the structure that I'm imposing to present these, and also for you to learn about these, is the following. So the basic question is always, what's the standard paradigm? Because once you know what the paradigm is, it's much clearer of what's going on. And so there's usually a specific context in which a respective ERP is elicited, and then there's a specific stimulus set. With the cognitive event-related potentials, what is quite often the case is that you can find this phenomena in all sensory modalities. For example, mismatch negativity is typically done in auditory stuff. There was just a paper by the Klaas and Stefan group from Zurich that looked into modeling of mismatch negativities in the visual domain. I'm actually reviewing a mismatch negativity visual paper right now, and I worked on with Felix on a somatosensory mismatch negativity. So they exist often over the sensory modalities, but the context then is important. And having said that, there's kind of the typical sensory domain, for example, for the mismatch negativity that used to be the auditory domain. And I'm still convinced that you get better mismatch negativities in the auditory domain than in the somatosensory domain, although I never done that myself. But the somatosensory mismatch negativity is such a filly thing, so I would rather have done some auditory stuff in my postdoc, but I do what the supervisor tells me to do. Then the other question is, once you know the paradigm, the mismatch, the cognitive event-related potentials have kind of their canonical electrodes. So it's not like for the ERPs that you see when you have 64 electrodes or maybe even 128 electrodes on the scalp, that you see the same effect everywhere, but they have spatial topography and also spatial localization in some cases inside the brain, which one should know about, so that one isn't surprised if one is actually trying to do an experiment building on, for example, the mismatch negativity, that one doesn't find it in certain electrodes. Then the bigger question, of course, is not which electrodes show these effects, but where do these effects actually come from? And this is kind of partly known, partly then not known, and active part of research. And then, there is kind of this typical neuropsychological story around these ERPs. And I try to dissociate this standard paradigm from this neuropsychological story because one thing is what actually happens, and the other thing is what psychologists then tell you what this all means. And it's good to know what some people think what all of that means, so that you have something that you can relate to, and then say, maybe I'm trying to give a quantitative description of this, so I'm making this neuropsychological theory quantitative. Or you can say, I don't believe this story, so I have another story, whatever. So keep this different. And these are the things that we want to then look at. So these four things: standard paradigm, electrode level effect, generating cortical systems, interpretation. We want to look at for each of these cognitive event-related potentials that I am discussing here. And we start with mismatch negativities. Any questions up to this point? No. So again, there will be slides that summarize the whole thing for you to have a reference that you can memorize, and then you have everything you need to know. Not everything, but you have a foundation of what you need to know to be able to talk to cognitive neuroscientists. And when they say mismatch negativity, you roughly have an idea of what they mean. And then there were slides with nice pictures. This is a slide with the content. Actually, it's from this Naatanen clinical neurophysiology review, and Naatanen, he's kind of the founding father of the mismatch negativity story. So he did EEG mismatch negativity primarily auditorily in the 60s and is still around. And so he's the authority for mismatch negativities. So the standard paradigm used to be, and most of it might still be, but there are also other paradigms now being increasingly used. The standard paradigm, nevertheless, is the oddball design, where you present, and we've seen that in the in previous lectures, where you present what people refer to as standards and deviants. standards are identical stimuli, and really the standard paradigm for mismatch negativity used to be the auditory mismatch negativity. So in this case, it would be a sequence of identical binaural auditory tones, which are referred to as standards. So people sit there, have some earplugs, have the EEG on, and have some earphones. Have some earphones and hear a tone. It's usually a pure sine tone, so it's like a doot, doot, doot. And it's presented quite fast, so more than every second. And sometimes there's a deviating tone. Yeah, so it's really like doot, 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 doot. That's about it. The important thing is that people don't, in the mismatch negativity paradigm, don't attend to the stimulus train. So they usually do a secondary task like watching a movie. Doesn't really sound like a task, but that's kind of a typical thing. So they are not actively processing the stimulus sequence by either overtly responding to it or covertly doing something with it, like counting how many deviants there are. They're not doing any of this. They focus on something else. And although the standard paradigm is the auditory, I told you before that somatosensory and visually domains also evoke similar things. The typical electrode level effect that one is interested in is a negative deflection for deviants versus standards. So it's about this comparison of a deviant versus a standard being defined, of course, by the context, standard, so by the fact that it's coming up most of the time. And this negative deflection is around 150 to 250 milliseconds post stimulus. And it's mainly found over frontocentral electrodes. Depends a little bit. This definitely holds for the auditory mismatch negativity under binaural stimulation. Then you definitely see this central pattern. So that's really fast. So that's 150 milliseconds after stimulus. And we will see how this relates to the neuropsychological theory. The cortical generating system, bilateral supratemporal process, this is kind of where also the auditory system sits. It's for the auditory mismatch negativity. This is mainly coming from the auditory cortex. There is some question about frontal processes. I will show you the data on which this is based, or at least the study, not really the data. And this people debate that. So people actually want that this is also coming from some kind of frontal source, because it should be a little bit more interesting if also frontal cortex is involved. My personal feeling is that this is a little bit of wishful thinking, so you can definitely get, you see these negativities in regions of the brain or regions of EEG that relate to the sensory cortices. Whether you actually need much of frontal stuff there and how strong the data supports that, I'm a little bit more skeptical, but we will see. And then the important thing is that because it's so fast, and because people don't attend to the stimulus, and because it's of course related also to the sensory areas, that this is an automatic, this is the mismatch activity is a reflection of an automatic pre-conscious processing of unexpected sensory input. So the idea is, and that's an interesting idea, that the brain is sensitive to the statistics of its sensory input. So as long as the statistics of the sensory input do not change, so you always have the same beep, the brain does not respond. But once there is something that deviates, so the statistics change, then the brain responds. So that and it does that without actually conscious effort. So that essentially the brain works as an unconscious statistical inference machine that detects changes. And this is also not that surprising because this is, of course, also what the retina or what any nerve cell essentially does. So if you stimulate something all the time, it will adapt, habituate, however you call it, and not respond anymore. And only if you stop responding, then you might get a omission response or something, or if something changes in terms of the input, then you get some activity. It's the same thing like for bar detectors in the visual cortex. As long as there's a white field, these things don't respond, these cells, but once there's a change in the input because there's an edge or something, then you respond. So the story is not that surprising, if you want, but relates to very common phenomena. So now a little bit more of a visualization of the whole thing. So this is from this Nathan review, and here you see kind of an old school way of presenting stuff, but at least I think no, it's completely wrong. So the mismatch negativity, so you see this is really old school. So positivity is plotted downwards, negativity is plotted upwards. And then you don't have like proper axis labeling on each of those, but you have one axis for all. In any case, you have here the stimulus onset, and you have two conditions. In green, you have the deviant, and in dashed black, you have the standard. And that's what's shown here. And you see that if the deviant deviates more from the standard in terms of its physical deviation, the mismatch negativity, which is the difference between the green line and the black line, and you have to adapt in this case to the idiosyncrasies of EEG researchers from 1950 to 1990 or whatever. You get more negativity after 150 seconds if you have a deviant. And this is shown for a single electrode. So there are two things that you see from this. So one thing is that this mismatch negativity is not like a on-off response, but it's kind of parametrically modulated by how deviant the deviation is. And you should also then already keep in mind, but isn't this like confounded by the stimulus? Because here you have the stimulus and the deviant, obviously they're different stimuli, so why is this not just the response to a high frequency tone, but why is this interpreted as a mismatch negativity? And that's something that you always, actually these kind of experiments, they don't take care of that. And also, there's another level to that. So on one hand, you have you look at different kinds of physical stimuli. So, for example, the standard 1000 hertz and the deviant here 1032 hertz. These are different stimuli. You observe two different responses. So how can you say that this response is really to the fact that this is surprising because it's a deviant rather than it being just a different stimulus? And you need to, ideally, you would control for that. So maybe one thing that you could do is that you have a standard of 1000 hertz, and then you take a deviant that is, let's say, 50 hertz above it, so 1050 hertz, and another deviant which is 50 hertz below it. So you have 950 hertz, and then you average over the higher deviant and the lower deviant, and then look at the response. So then at least you have averaged over both the higher and the lower deviant. Another way to do that is as a deviant, actually use an omission, so don't provide a stimulus at all. So then the deviant, at least you cannot say it's due to the physical properties of the stimulus because it's not existing. Yeah, in other ways that's essentially about the stimulus. There's another problem with these typical experiments that use 20%, 80%, that if you average the data for the standard and you do like 1,000 trials, then obviously you have 800 trials for the standard and 200 trials for the deviant. And then, of course, you can estimate the ERP curve for the standard much better and with less error than for the deviant. And what I typically find is, although that's not necessarily the case, is that the standard curve, or essentially the curve that you get from averaging more trials, usually has a shallower profile than the one that you get from having less trials. So ideally, you first of all, if you really want to study mismatch negativity, you account for the fact, of course, you have to induce a change, but you should make sure that this change and physical stimulus properties are not confounded, in the sense that whenever there's a change, the stimulus changes in a pre-specified direction. So it's always, for example, louder or of a higher frequency. And the other thing is that if you compute the ERPs, you should also make sure that you have the same number of trials, so that your estimation quality for both ERPs is the same. And of course, this has been done, but not necessarily all the time, or not necessarily is done all the time in the work that is being done now. So, if one wants to talk about mismatch negativity, one should actually take care of these things, which might also then result in the fact that your mismatch negativities might not look as large as here. So this is just the difference curve between the standard and the deviant, but smaller. And then you still need to believe that this is a mismatch negativity, although it's not as strong as you would have supposed, as you would have gotten if you used a less well-controlled experiment. Questions about this? No. Good. This then shows an example of an attempt for source localization. So this is auditory mismatch negativity. Here you see the topology. So that's the difference. So essentially you can think about that this is the difference between deviant and standard that's where it's at 160 milliseconds post stimulus, a little bit later and a little bit later. That must be wrong here. That's maybe 200. It's definitely not 167. Maybe it's 170. Something funky here. It doesn't go back in time. And this is where this was localized to. So this, of course, depends on how you do the source reconstruction, and we will talk about source reconstruction. But this is kind of the evidence where people always say it's also a frontal source. So first is here kind of coming basically from where auditory cortex is sitting. So this is trying to explain this. And then a little bit later, there seems to be a little bit of a frontal shift. Yeah, I see that this is a little bit more frontal and there's a little bit less here, but it's not that it's like here frontal wise, and it's more like a weighting that's a little bit stronger up here. So that's where the story about the supratemporal and frontal sources essentially comes from. For auditory mismatch negativity, now yours truly worked on the somatosensory mismatch negativity, which was described in a couple of papers by people from Japan prior to our work here. But there were like 4 papers or something. So it was not, so in auditory mismatch negativity, there are quite a number of them. So what this shows are electrode level effects, and then also Topple plots. And here you see deviant and standard. Here, deviant and standard are actually estimated from the same number of trials and averaged over a higher amplitude deviation and a lower amplitude deviation. So this was based on a roving paradigm, which means that you have trains of stimuli of low amplitude, so sorry, that was somatosensory, tactile, electrical stimulation, and you have stimuli of a certain amplitude, so ampere, and then a higher amplitude, and there were always like blocks of 8 or 16 stimuli of the same amplitude and then of a different amplitude, and then the deviant and the standard are always the ones around the switch. So deviant can be high and low, and a standard can be high and low amplitude. And one can also plot this for the high and low and the deviant and standard, and gets a similar pattern in terms of the difference. This is average over both of them. And what I found there, and I actually started to call the whole thing more the mismatch response. Because actually in this paradigm, that's how sometimes things go. I think Felix was interested in the mismatch negativity, and then the postdoc who started this with a master student before I joined the lab and then did the basic modeling of that, they were also interested in P300. So we had this thing that people were actually attending to the stimulus. So this was not a mismatch negativity paradigm, but Felix was all about the mismatch negativity. And I then later called the whole thing the mismatch response, because we also have P300 effects there. In any case, in the temporal ballpark of the typical mismatch negativity, so 150 to 250 milliseconds post-stimulus onset, we see this negativity primarily over secondary somatosensory cortices. And then we have more later P3A and P3B responses, but there are also differences a little bit earlier on. So this is why I later called this whole thing the mismatch response, not the mismatch negativity, because people get quite sensitive about these things. So if you say I have a mismatch negativity, then everybody has in their mind 150 milliseconds to 250 milliseconds. And then if you say, oh, look here, I have a mismatch negativity, and then they will be like, wow, that's 85 seconds. That cannot be a mismatch negativity. That's something else. You did something horribly wrong. So that's when it comes to these kind of terms, although they are often quite fuzzy, people get really strong emotions about this. Because they have this thing that an effect occurs at a specific time in a specific region. It's always like that. It's always been like that. And if you look at the whole data and you look at all electrodes and over all the time, you see that there are differences, stronger and weaker ones all over the place, or maybe not that strong in somewhere and so on. And but you should always assume that there's somebody in the audience for whom a mismatch negativity is like 150 to 250 milliseconds, or a P300 is at 300 milliseconds. And if you show something that is like the P100 but a little bit late or something, they will say, no, that's not a P300 because it's not at 300 milliseconds. It happens. So be aware that if you do EEG research, that people expect the typical effects to be there when they should be there. Until you get this high-profile paper that says, for years, we thought of the mismatch negativity as something at 150 milliseconds. But now if one looks over the whole time point, then you see effects everywhere. And then everybody's like, wow, that's amazing. And then you have this highly cited paper which stated the obvious, but yeah, at least then open up things. Okay, so that was 2 meta. Yeah, that's the smarter sensory mismatching activity. Good questions about this. So learn these things. So that's my advice. Learn these things. Memorize them so that you know what's going on, and then relax. And if somebody says, here I have computer mismatching activity, and I say, well, typically the mismatching activity is 150 milliseconds. So can you explain how this relates to the mismatching activity? So that's more, life is nicer if you do that. Good. Mismatching activity, something unconscious that changes. P300, the main difference is that to mismatching activity, the paradigm in terms of that you do some active conscious processing, which can go to a certain step or go even that far that you respond to the stimulus. So this is a nice review about the P300. The standard paradigm for the P300 is again an oddball paradigm, usually auditory or visual. So P300 is actually, I think, also quite visual. It's presented frequency of around 1 Hz with occasionally deviating stimulus. That's so far they're identical to the mismatching activity. But then the important thing is that the participants are instructed to mentally or physically respond to the deviant stimulus. So people not watching a movie, but do some cognitive processing of the stimulus material. And then, of course, you can have variations and different expressions of the P300. So the electrode level effect, it's a positivity. It's later than the mismatching activity, so it's between, let's say, 250 and 500 milliseconds per stimulus, primarily over midline electrodes, so FZ, CZ, PZ are typical electrodes that one looks at. It's typically larger in amplitude in parietal compared to frontal electrodes, so there's somehow an association of P300 with parietal cortex. More or less. Then there are 2 subcomponents at least, and of course people like to find more subcomponents. The first one being the P3A, which is a short latency response to the infrequent distinct tone happening without associated tasks, so going more into the thing about mismatching negativities. That's why I said that this thing here is more like a P3A. What you see here, this thing that we then have in the very end, that's definitely a P3B. And this is more like a P3A compared to this. Anyway, the P3B then being the P300 proper, which is task associated and occurring at a longer latency, so later after the stimulus than the P3A. The cortical generating system is essentially the brain, so it's a parietal temporal frontal network. It's almost the entire brain. Then, of course, the interpretation is that it's novelty processing, and it's task related, at least for the P300 proper or the later response. So some people only call the P3B the P300 and P3A not, but yeah, I think it's helpful to have then a review article that establishes some common ground. Yeah.
So what's the difference then between P3A and mismatching?
It's later.
Just the latency.
Yeah. And that, and I'm not sure how much of a P3A you actually see if you don't. So it says without associated tasks, but if you, I'm not sure right now how much this task means a physical response. I mean, we'll see a P3A for no response, but I think the P3A might also be almost abolished if you don't do anything. So in the typical mismatch paradigm, let's look. I mean, we can look. It's the easiest. Well, there was a little bit of a P3A, right? But it's not that strong. So here, this positivity, that's a difference between the stimuli, is there in a mismatch negativity paradigm. But it's not. So this is, I mean, again, this actually speaks to my nomenclature, because so instead of calling here this is the mismatch negativity and fight for its latency and so on, why not say, okay, there's a mismatch response because they also have this positivity here. Then, of course, the other thing that you have to keep in mind is that if people look at these ERPs, they are also thinking about how fast are people in terms of response times. And then, of course, the P3A falls into something where people are actually able to respond. So people are definitely not able to respond to a stimulus within 150 milliseconds. That's too fast. The fastest response times that you get are like 300 milliseconds. And so this is why, just because of latency, you can say this is something not task related and so on. And P3A, although you might see it in a mismatch negativity, it's in some cases it might be task related, and people can respond at that time. I try not to base my. Anyway, that's a different story. I don't want to tell that story now. So let's look a little bit more into P3A, P3B business. So here, I always have to remind myself how that works. So here, first, we have a single stimulus being presented fairly occasionally, and people are required to respond to that. Then, without any access in this case, you get a P300, so you get a positive deflection around 300, 400, 500 milliseconds. So that's something you actually that's very reliable. That's something that you see all the time. Now, if you have a stimulus, you have a standard and a target, and you're required to respond to the target, then you get this big difference. Of course, there are also big differences in what's happening, because to the stimulus you're not pressing a button, but to the target you're pressing a button. Yeah. So you actually have a motor response that's going on here, which is the typical thing that is associated with P300, and you have the fact that you're responding to the deviance. Yeah. Actively by pressing a button. And what is then shown here is that you have a standard stimulus to which you're not responding, you have a target stimulus to which you're responding, and you have a distractor stimulus to which you're also not responding physically, so you're not pressing a button, but you're of course attending to it, because you have to attend to the whole thing to be able to press the button when you see the target stimulus or hear the target stimulus. And this is then what is shown here, that the P3A, which is quite big, is the response to the distractor stimulus, and the P3B is the response to the target stimulus when you press the button. So the distractor elicits a P3A and target elicits a P3B. And you can, of course, then try to, I'm not sure whether they did that, maybe I guess, you can subtract from something like this, you can subtract the response to the distractor stimulus. Ah, yeah. This is stupid Windows. So the good thing is you can watch it on the video. Then after the fact, you can understand what I meant. because there you see it. So I'm not quite sure, but I would assume, yeah, no, it looks strongly like that. The response to the target stimulus is this thing, and then if you subtract from it the response to the distractor stimulus, then this list is P3B. So I'm not completely sure how they came up with this trace here, but it looks strongly as if they did some differences. The important thing is that for the distractor stimulus, you get this early thing, and that the P3B is more related to the actual button press. Yeah? So that's kind of the general logic. So it also makes some kind of sense if you think of it from a cognitive perspective. So first you realize that something is deviating, and then you press the button, and this then is dissociated in the ERP components. So, I tried to figure it out myself, but it was actually on the slide how the whole thing worked. So the target stimulus does the P3B and the distractor the P3A. So cortical generators, this is from somebody who always sends me stuff from in Neuroimage to review, Tom Eichele, who's in Bergen. So that's his work in the 2000s on the P300, where they did some EEG-fMRI, and then they did some EEG-fMRI correlations, and without going into the details of these figures, going over these different peaks and looking into the correlation also with the bold signal, a lot of brain regions show up. So it's this frontal, parietal, temporal network, which is almost the whole brain. Yeah, and that's kind of at the level where we can keep that here. Of course, you will find papers that argue that P300 is primarily a frontal business or primarily a parietal business, and then you can look at how they conclude that. But yeah, you should not be surprised if you're trying to do source localization, for example, on a P300. Which essentially, depending on how you stimulate, but it looks like the topography is fairly unspecific. So the topography, I don't think I have a plot of the topography, but it's also, it looks pretty much like this, so that you have basically you have a central positivity that extends over the whole thing, and then if you do social construction, you cannot really hope that you will find something very fine-grained in terms of the cortical response, like as you would if you have a nice dipolar pattern, which has like we've seen last time, where you can, if you think about it, if you put a dipole into the brain, there's actually only this dipole that explains that. If you have this overall positivity, then of course it will not be like a nice dipole somewhere, but it will be everything essentially contributes to that. Good. Let's have a break now and then talk about language-related event-related potentials. Are there any questions about the P300? Mismatch activity versus P300. So you should definitely know what the paradigm difference is between a mismatching activity and a P300, so that, unlike me, you don't come into a lab where they want to study mismatching activity, but use a paradigm that's actually more P300 related, and you can take care of that. Okay, then let's have a break and then talk about language-related potentials.